This is where you are. So, into the camera coming along. Okay. It's Raghavan. It is Raghavan. Good morning, Dr. Raghavan.
Good morning, Bhaskar.
Hi, Dr. Raghavan. How are you?
Hi. Long time no see.
I know. Where are you? Hyderabad?
No, Bangalore.
Bangalore. Okay.
I moved to Bangalore a couple of months back.
Moved
September.
means
I moved to Bangalore in September
moved for good.
for good.
Okay. Okay, good. That's you. One of the girls is there?
Nobody is here actually. That girl who was here, she has gone to Singapore.
Okay. Okay.
But here, I have an apartment here, so I moved into that apartment.
Oh, that's good. That's good. Anyway, it's a nice city to
It's
be
a
in.
nicer place than Hyderabad. Hyderabad
Yeah,
is-
and we have a temptation because we can see you again because Bangalore, we come once a year,
Mhm.
at least on our way to Palghat for the family puja.
Mhm.
This year we couldn't make it, but we do. Bangalore is, we come.
Okay.
So
When you come
we
next
can-
time, I will send you my contact.
Okay.
we'll call you for sure before we come.
Yeah.
But anyway, today, I mean, these orations always, Dr. Raghavan, the person who's getting the award today,
Mhm.
we sent our daughter the link and said that this oration is taking place, etc. When she saw the name,
Mm.
she realized that she had met this person in Stanford when she
I
was
see.
doing her postdoc there. And they talked they dealt with each other professionally and they clicked and she had her contact, but she sent her a mail and said that, this is my grandmother.
Mhm.
And so that she was quite, she said, oh, really? Because they never talked of family.
Everybody is hearing this.
Yeah.
Entire, all over the world. So,
Bhaskar says not to give such details because the whole world is listening.
That's true.
I'm sorry, I thought I was talking to you.
We are public now.
No, I'm very low tech on all this, so I thought I was talking to you. Sorry.
No. I think we can have a long chat.
Yeah. No, Nitya was going to try and get onto online towards the end because of the time difference.
Yeah.
So let's see if she makes it. On the,
I see you have about 10 people logged in.
I have communicated to as many people as possible who know Dr. Devi, who were there at PGI at that time. So hopefully some of them would join.
yeah.
Sarla also is doing the same thing.
Yeah,
Yes,
we
ma'am.
spoke to her. We had long chats with her yesterday.
Mm.
Yeah.
ब्रोशर वर आहे बघा ब्रोशर व्हॉट्सअपवर
Good morning, all. Hello. Hello. Good morning. Please settle down. Good morning. A very warm welcome as we begin the 32nd Professor P.K. Devi Memorial Oration Award presented by ICMR, National Institute for Research in Reproductive and Child Health, Mumbai, and Kasturba Health Society Medical Research Center, Mumbai, to Professor Dr. Gagandeep Kang, who is Director, Enterics, Diagnostics, Genomics, and Epidemiology, Global Health at Bill and Melinda Gates Foundation, in appreciation of a pathway-breaking achievements in the field of infectious diseases. On behalf of ICMR, NIRCH, and KHSMRC, I thank Dr. Gagandeep Kang, who had agreed to deliver the oration on the title, Building Human Capital in India, Cohort Studies, Nutrition, and Need for Collaborative Science. We are indeed honored to have Dr. Vandana Patravale as a professor of Pharmaceutics, Institute of Chemical Technology, Mumbai, India, who has graciously agreed to be the chief guest for the event. We welcome you, ma'am. And also, Deepak has now joined. So we begin the ceremony. For that, I would like to invite Dr. Shobha Iyer, ma'am. Please welcome Dr. Shobha Iyer, who's Assistant Academic Director, Medical Research Center, Kasturba Health Society, who will lead us in the lamp lighting and invocation for today's event. I would also like to invite our Director, ma'am, Dr. Gitanjali Sachdevan, ma'am, our ex-director and now emeritus scientist, ma'am, Dr. Smita Male, ma'am, Dr. Vandana Patravale, our chief guest, ma'am, Dr. Jayashree Joshi, honorary consultant and emeritus professor, KHSMRC, Dr. Shobha Devi, ma'am, research director and head medical research center, Kasturba Health Society, Dr. Tina Joshi, ma'am, scientist and head of application and implementation research, and Dr. Ranushi Patil, scientist C and head of department of clinical research, to come on the stage for lighting of the room. Unfortunately, Dr. Rama Vaidya, ma'am, who's the Honorable Director and Division of Endocrine and Metabolic Disorders, Kasturba Health Society, could now join us as she is unwell. Probably she is trying to join us online. Please come on the stage for the lamp lighting. वक्रतुंड महाकाय सूर्यकोटि समप्रभ निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा या कुन्देन्दु तुषारहार धवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिभिर्देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा। मैत्रीं भजत अखिलहृजेत्री मैत्रीं भजत अखिलहृजेत्री
मैत्रीं भजत अखिलहृजेत्री आत्मवदेव पराणपि पश्यत आत्मवदेव पराणपि पश्यत युद्धं त्यजत स्पर्धां त्यजत युद्धं त्यजत स्पर्धां त्यजत त्यजत परेष्वक्रममाक्रमणम् त्यजत परेष्वक्रममाक्रमणम् मैत्रीं भजत अखिलहृजेत्री मैत्रीं भजत अखिलहृजेत्री जननी पृथ्वी कामदुघास्ते जननी पृथ्वी कामदुघास्ते जनको देवः सकलदयालु जनको देवः सकलदयालु दाम्यतदात दयत्वं जनतां दाम्यतदात दयत्वं जनतां श्रेयो भूयात् सकलजनानां श्रेयो भूयात् सकलजनानां श्रेयो भूयात् सकलजनानां श्रेयो भूयात् सकलजनानां श्रेयो भूयात् सकलजनानां
Thank you, ma'am. Now I would like to invite our director, ma'am, Dr. Xxxii Prof, for this special occasion to deliver the welcome address.
So, very good morning. On behalf of the National Institute for Research in Reproductive and Child Health and Kasturba Health Society Medical Research Center, it's my privilege and pleasure to welcome you all to this Professor P.K. Devi Oration Award ceremony. First of all, I would like to extend very, very warm welcome to the star of today's event, Professor Dr. Gagandeep Kang. Her first name is, literally it means that star, light of the sky. And she has really touched the sky of glories and made all of us, all Indians, very, very proud. So thank you, Dr. Kang, for accepting our invitation. I would also like to welcome our chief guest, Dr. Vandana Patravale, very distinguished professor from ICT Mumbai. Thank you, ma'am, for being with us today. Thank you so much. As you know that we all began this year by paying our tributes to our founder director, Dr. Shanta Rao. And as it was her birth centenary year, and now we are concluding this year, almost concluding this year, by celebrating another great women scientist, Professor P.K. Devi. She was our acting director and academic head during 74-75. But her contributions were not really limited to our institute. Those contributions went beyond our institute. In fact, she was instrumental in establishing many OBGY departments in several medical colleges of India. She was the one who could foresee the relevance of clinical research and the role of medical colleges in inculcating the spirit of research among medical students. And many years down the line, we are still reaping the benefits of her vision. So to celebrate her accomplishments and remember her very fondly, we had instituted this Professor P.K. Devi Oration in 1992. Since then, every year, probably except 2020, we have many distinguished personalities, including scientists, policymakers, and also clinicians who have been delivering this prestigious oration and sharing their perspectives and pearls of wisdom. And for this year, we have Professor Gagandeep Kang. You all know about her. I really don't need to speak about in detail. You know that, you know her key role in the development of indigenous rotavirus vaccines and her path-breaking work in enteric infections and their sequelae in India. I would just say that in Dr. Kang, we have a great role model for us as well as many young girls sitting in the audience. So I assume that it's really late evening in the US from where she's joining. So I won't really stand long, but before I leave the floor, I would like to thank Dr. Ashok Vedya, Dr. Rama Vedya, and Dr. Smita Mahale, our past director, for her inputs and support to this event. I would also like to place on record efforts taken in by my colleagues, Dr. Anushri Patil, Dr. Dipti Tandon, Dr. Beena Joshi, and their teams, and also Mr. Kohli and Mr. Lobo for putting together this event. So thank you all, and I wish you all two very great, enjoyable, and inspiring novels. Thank you so much.
Thank you, ma'am. We are also thankful to you for all the help you have extended, which has enabled us to conduct this event. As we move on, let us extend our warm welcome to Dr. Shobhavati P. ma'am, who is the Research Director and Head Medical Research Center, Kasturba Health Society, who will introduce our distinguished chief guest.
Good morning, everyone. It is my honor and privilege to introduce Professor Vandana Patravali. She is a distinguished professor of pharmaceutics at the Institute of Chemical Technology in Mumbai. She has had a very remarkable academic and research career over the last 30 years, numerous publications and so many patents. She is indeed a prolific scholar and a very innovative inventor in the field of pharmaceutical sciences. And I'm very proud to say that another role model, because she's listed consistently in the top 2% of world scientists in pharmacy by Stanford University, USA. She has diverse research interests, which include nanocarrier development for infectious diseases in the areas of cancer and neurodegenerative diseases. And she has also worked on inventing medical devices, nanodiagnostics, and nanovaccines. She is globally recognized and has been awarded many awards. For example, the Most Iconic Healthcare Leader Award globally in 2023, very recently. The Abdul Kalam Technology Innovation National Fellowship in 2021 and Fellowship of the Indian Chemical Society in 2020. She is very actively involved in teaching. She has leadership roles like being the president of the CRS Indian chapter and is extensively engaged with industry, highlighting the profound impact her work and contributions have on healthcare and innovation. There has been a transfer of more than 20 technologies to industry, which makes her contributions to this field and to our country very, very valuable. And her influential editorial contributions to peer-reviewed journals and newsletters is the last which I will mention, but it's not the least. Thank you very much, madam.
We are honored to have Dr. Vandana V. Patravale as our chief guest today. I request ma'am to kindly address the gathering.
Thank you, Dr. Shobha, for those kind words. My pranams to Dr. Ashok Vaidya, Dr. Rama Vaidya. Namaste and good morning, dear all. It is an absolute privilege, I would say, an honor for me to address this distinguished gathering. And I'm really humble standing before all of you. In fact, generally, I take only technical lectures, but when Dr. Rama Vaidya called me up, it is only due to sheer respect and affection for Vaidya's families that I said yes. Thank you, Dr. Rama Vaidya. I would also extend my heartfelt gratitude to the organizers, the ICMR National Institute for Research in Reproduction and Child Healthcare, as well as Kasturba Health Society's Medical Research Center, Mumbai. Dear friends, we are here to commemorate the illustrious Professor P.K. Devi, a clinician, researcher, scientist, administrator, builder of institutions, a legend. And we are really privileged to have with us Professor Dr. Gagandeep Kang, the first Indian woman elected as Fellow of Royal Society of London, whose work, as mentioned by the director, in Enterics, Diagnostics, Genomics, Epidemiology has been instrumental in shaping the landscape of healthcare, not only in India, but worldwide. A living legend, indeed. Salute to you, madam, and welcome to this Professor P.K. Devi lecture. Truly, it is an honor to listen to you on a topic that holds paramount importance for the future of our nation, building human capital in India, cohort studies, nutrition, and the need for collaborative science. Now, being professor, I can talk for 45 minutes, but in the limited time which I have to do the justice, I would read the thoughts which I have penned down. In this era of rapid advancements and global challenges, the significance of investing in our human capital cannot be overrated. Our nation is progressing, and in fact, its prosperity is inherently tied to the health, education, and well-being of people. So it's time to truly acknowledge and act. Also, it is increasingly evident that robust cohort studies are the cornerstone for informed decision-making and evidence-based policies. These provide invaluable insights into understanding the complexities of human health and development across various life stages. They enable us to identify critical factors influencing health outcomes, thereby guiding interventions that have lasting impact on society. Nutrition, on the other hand, stands very tall as a fundamental pillar in nurturing a healthy populace. Adequate nutrition during the formative years not only ensures physical growth, but also plays a pivotal role in cognitive development and long-term health. The role of microbiota, and especially gut health, to overall well-being is being studied much in detail. And I'm sure even here in this institute, a lot of work is being done on the same. Though, of course, our ancient practices spelled it centuries ago. It is imperative that we prioritize efforts to address malnutrition and its multifaceted implications. Also, we cannot neglect overnutrition and its causative factors which it can have on diseases. So I'm sure our esteemed speaker will talk at length about this. Collaborative sciences stands as a beacon of hope in our quest for progress. The collective synergy of researchers, institutes, of course, the policymakers, and community at large is indispensable in addressing complex health challenges. Collaborations foster innovation, facilitate exchange of ideas, and accelerate the transition of research findings into tangible solutions that benefit society at large. I'm really glad that with this institute, with Dr. Smita Mahale, Dr. Venno, Dr. Vikrant, earlier Dr. Vanage, Dr. Dige, all of them, we have been collaborating, and even with Kasturba Medical Center, we have collaborated. Need of ours is to create, of course, an ecosystem that nurtures collaborative efforts, fosters interdisciplinary research, and promotes knowledge sharing. Our new education policy 2020, if you have read, also has duly considered this. And therefore, many colleges and institutes are establishing contacts with other universities to have multidisciplinary centers. The very first Professor P.K. Devi oration in 1992 was by Professor Bergstrom, a Nobel laureate. I happened to see its recording, in fact, where Dr. Rama was talking. And this point of collaborative efforts was even stressed in that very first Professor P.K.
He's
Devi oration.
introducing the speaker.
The point then is, what are we missing? Is it that we are reluctant to initiate a dialogue? Or is it that we lack mutual trust? Or is it that we have very big ego? What I'm trying to say here is that we've gone a full circle talking about collaborative sciences, but yet the transition because of the collaborative efforts to industry is not happening. So we need to take a lead. We need to go talk to people. We cannot just sit idle and think that people will come to you. Of course, KHSS, Medical and Research Center, is a perfect example of collaboration, I would say. Kudos to the vision of pragmatic leaders who have embraced team spirit. That's the only center where I've seen medical practitioners, Ayurvedic practitioners, research scientists, technicians, everyone working together, not only nationally, internationally, networking and moving ahead. What I still need to see and I'm longing to see is how many technologies based on that basic research which is carried there have gone in the market. And there again comes the collaborative efforts. I also commend the remarkable work undertaken by ICMR, Bill and Melinda Gates Foundation, and countless other organizations, I would say, and individuals who have dedicated to advancing global health. Their unwavering commitment and tireless efforts have significantly contributed to shaping the landscape of public health. Our institute, ICT, being technology-driven, definitely contributes in this journey too. We have about 300 first-generation entrepreneurs, and that's the reason, I would say that they keep coming back and we keep doing better. However, still lots needs to be done. As we move forward, let us pledge to champion the cause of building human capital in India and worldwide. Let us collaborate, innovate, and relentlessly strive to create healthier, more prosperous future for generations to come. In conclusion, let us honor the memory of Professor P.K. Devi by continuing to strive for excellence, by fostering collaboration, and by dedicating ourselves to the cause of building a healthier, more prosperous future for India. Thank you. Jai Hind.
Thank you, ma'am, for such an enlightening welcome address. So our next segment celebrates the life and contribution of Professor P.K. Devi. To share more about this legendary figure, I invite Dr. Xxxii Joshi, who is an honorary consultant and emeritus professor at KHSMRC and former deputy director at ICMR NIRL, to the stage.
Can I put it down a little? Because they will feel otherwise as if it's an Akashvani. Yeah. No, because people can't see me from there. It's okay. 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 Thank you, Dr. Deepti. Good morning, Dr. Gagandeep Kang. I don't see her here just now, yeah, but it's okay. Yeah, we have seen her earlier. She is joined. Dr. Vandana Patravale, Dr. Ashok Vaidya sir, Dr. Rama Vaidya sir. I hope they have joined online, but I can't see. I don't know whether someone from Vardha also has joined. Dr. Gitanjali Sachdev, Dr. Shobha Udupi, Dr. Smita Mahale, Anushri Patil, Dr. Ashwini Kumar Rao, Dr. Beena Joshi, Nutan Nabar, Dr. Bhaskar Balkrishnan, Dr. Devi's son, who has joined us. Dr. Shobhana, Dr. Lakshmi Mehta, our previous orator. She is Dr. Devi's daughter, and she also had said that she'll try to join. Other dignitaries, friends, students, and all invitees. It's a great honor to introduce Professor P.K. Devi, indeed a legend, in whose memory we are holding this oration. Not that she's unknown. Some of you, particularly those who are not there in the hall, have been associated with her, but it is necessary to recapitulate the highlights of her inspiring life work for all of us, particularly for those who have not known her closely. Welcome, Dr. Gagandeep Kang. I had heard your views earlier in the ICMR DBT webinar on ethics in clinical trials for infectious diseases and studies in human volunteers during the COVID pandemic. And now I have the opportunity to hear your views on cohort studies, nutrition, and need for collaborative science. It is an occasion to be marked because we have Professor Vandana Patravale, the expert in pharmaceutics, as our chief guest. Institutional collaboration is an essential component of most aspects of research. The development of the new male contraceptive and the COVID example are recent examples. And I'm sure collaborative research will pave the way for new discoveries for both NIRRCH and KHSMRC. Professor Davis' surgical skills, teaching abilities have been experienced and described by many. Her formal biodata is also there on the invite. However, today in the few minutes allotted to me, I would like to describe her as a clinical scientist, an able administrator, and a human physician. A few things may be repetitive because some things have been described by the earlier speakers also. But it's worthwhile repeating because if you want to learn, there's nothing like repetition and learning and absorption. Her knowledge and discipline were not restricted to the OPDs, wards, and operation theaters. She had a profound understanding of the basic sciences, reproductive endocrinology of men and women, and collateral sciences like biochemistry, pathology, pharmacology, and preventive medicine. She respected and also collaborated with these departments, and many papers are available on the internet. Her contribution to the development of 3-monthly injectable spacing method, DMPA, in the Upjohn company in US, later also culminated in WHO ICMR clinical trials, not only on DMPA, but many other injectables, oral contraceptives, intrauterine devices, chemical sterilization. And I have participated in some of these, including vinagrin chemical sterilization. Her contribution to research on different types, doses, and drug delivery systems of prostaglandins for induction of labor and induction of medical abortion and for control of postpartum and post-abortal bleeding was significant. After her relatively premature demise, the Acta Obstetrica et Gynecologica Scandinavia brought out a special issue dedicated to her memory and work in 1988. This field has continued to develop further until now, and recently there was a news just last week that misoprostol alone is effective as a single drug for inducing medical abortions. Her advocacy for legalization of safe medical abortions in India is well known. And whilst this is still elusive to many women in some developed countries even today, without doubt, this has contributed to a reduced rate of criminal unsafe abortions and maternal mortality, and indirectly also reduced anemia. Madam Udupi, she is also fighting against anemia, a chronic enemy of women to say the least. She also investigated several other clinical problems like toxemia of pregnancy, pregnancy, or that is same as PIH, pregnancy-induced hypertension, male-female infertility, and other problems. She also strengthened the area of genetics at this ICMR institute. Her interest in science extended beyond the conventional medicine. Her basic knowledge of Sanskrit and Ayurveda propelled her to conduct some studies on Ayurvedic products while she was in IRR as the administrative head, and she collaborated with the various departments. Believe me, for the first time in medical education, after my MD and after my initial ICMR research posts, I was introduced to Ayurveda and Charak during her tenure. I had the opportunity to participate under her guidance in a study on an Ayurvedic contraceptive, and we studied the possible mechanisms of action in healthy women, and also that was after the first ethics committee that was formed, and I used to be the member secretary at IRR, so it was all with the ethical consent and all those preliminaries. And we studied the history, clinical examination, pap smear, not only for exclusion of cancer, but also for hormonal cytology, cervical mucus, postcoital tests, endometrial histology, and serum progesterone levels. Sonography was restricted to obstetric diagnosis mainly those days. This no doubt prepared me for the phase 1 to 3 clinical trials on long-acting injectables and implants later on. I remember actually as an undergraduate, I had a very short time with her. And during my first house job in Nagpur, she was not, maybe she was on leave, I think. And I didn't interact much with her. But as a student and as an intern, I remember that she made us do the daily urine test ourselves for cases of preeclampsia, for the proteinuria. And we were told not to depend on the nurses or ayahs to do the same. Her encouragement for the initiation of a separate endocrinology and infertility section at NIRRCH under the able guidance and leadership of Dr. Rama Vaidya was recognized by Dr. Rama Vaidya and her senior colleagues, including others, with the institution of this series of annual orations. We all know that both Rama Madam and Vaidya Sir have been associated with the inception and development of Women's Park and current KHS MRC. And Vaidya Sir's ancient and novel integrative insights in conventional and traditional medicine have been the foundation of our center. Her great administrative capabilities are evident from the systematic establishment of undergraduate and postgraduate courses in the medical colleges, as has been mentioned, along with other senior colleagues in the ICMR and WHO. She also strengthened the National Network for Human reproduction research centers, or HRRCs as they are known. And now they still continue to participate in multicentric studies of the ICMR or WHO. There are various topics, I mean, contraceptives or endocrinology or whatever it may be. She was a strict disciplinarian, and her classroom or OT doors would be closed for latecomers. The lady students, luckily, boys didn't grow long hair those days, had to tie up their hair tidily in the OT and wear a cap. Otherwise, they were at the risk of being at the other end of the scissors. Nevertheless, she was soft-spoken, had a great sense of humor, and was always smiling. And these qualities made her a more efficient yet popular administrator. Her concern for patients was holistic and not disease-centric, as she could identify the social factors which aggravated diseases. Her work in prostaglandins has formed the basis of national network in primary health care for management of labor. Indeed, her empathy extended beyond routine care, and she reserved few beds in her ward to look after unwed teenage mothers and facilitate adoption of their babies. At one of the PK Devi orations at our center, she was paid a rare tribute through a poem by a Sahitya Academy Award winner, none other than late Sri Nandini Ben Mehta. And the title is summarized as "Maa ko bhi jiski god mile," meaning even the mothers found motherly love in her. Lastly, I would like to end my tribute to her by a slight modification, that is change in vibhakti or case as is known in English grammar, from the seventh case, that is bhuteshu, meaning within all beings, to the fifth case, that is bhutebhya, meaning for all beings. From the well-known Sanskrit saying, Ya Devi, sarvabhutebhya matrurupena, prajnarupena, karunarupena, urjarupena, samstita, namastasyai, 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 namo namah. Thank you.
Thank you, ma'am. Such an elaborate and a personal introduction you have given for Professor P.K. Devi. So as we move on, let's learn more about our orator, Dr. Gagandeep Kang, who has joined us online, and she will soon grace us with her insights. And to introduce her, I welcome our head of department, Dr. Anushi Patil.
A very good morning to all our dignitaries, all the invitees for the oration, dear colleagues and friends. It is indeed an honor for us to have the esteemed presence of Professor Dr. Gagandeep Kang to deliver this prestigious Professor Dr. P.K. Devi oration. Dr. Kang needs no introduction to this august gathering, but I consider this my privilege to do my humble duty of introducing our honorable orator, Professor Dr. Gagandeep Kang. Professor Kang is a renowned microbiologist and virologist and is currently Director, Enterics, Diagnostics, Genomics, and Epidemiology, Global Health, Bill and Melinda Gates Foundation. She was former Executive Director of the Translational Health Science and Technology Institute, known as THSTI. With an academic background of serving as an adjunct professor at the esteemed Christian Medical College, Vellore, India, Professor Kang's expertise centers on enteric infections and their impact on Indian children. Her groundbreaking work on diarrhea and rotavirus vaccines has had a lasting impact. Professor Kang chaired the World Health Organization Southeast Asia Regional Immunization Technical Advisory Group from 2013 to 2022 and was a member of the working group on COVID-19 vaccines established by the Strategic Advisory Group of Experts at the WHO from 2019 to 2022. She has contributed significantly to research, vaccine development, and public health by establishing national surveillance networks and supporting vaccine trials, resulting in 2 WHO pre-qualified vaccines. She has also contributed in scientific advisory committees of many Indian and international organizations. Her research explores the complex relationships between infection, gut function, and physical and cognitive development while striving to strengthen human immunology research in India as well. Professor Kang is a recipient of numerous accolades, including the Infosys Prize in Life Sciences in 2016 and a distinction of being the first Indian woman elected as a Fellow of the Royal Society in 2019. She is also a member of the US National Academy of Medicine since 2022. Her active involvement in various Indian and international committees such as CEPI, WHO, and the Gates Foundation highlights her dedication to global health. Furthermore, she has nurtured the growth of a new generation of clinical researchers in India by establishing training programs in clinical translational medicine at the Christian Medical College, Vellore. Professor Dr. Gagandeep Kang's remarkable contributions continue to shape the landscape of global health research and policy. It is indeed a pleasure to have such a luminous stalwart for today's oration. We welcome you, Madam, and invite you to deliver the oration for this August gathering, which will be preceded by the citation reading. Thank you.
Thank you, ma'am. I now welcome Dr. Smita Mahale, ma'am, who is our ex-director and emeritus scientist for the citation reading, setting the stage for our distinguished speaker. Welcome.
Thank you, Dipti. Good morning to one and all. Good evening to Dr. Kang. It's my great pleasure and honor to read the citation for the 32nd Professor P.K. Devi Memorial Oration Award, presented by ICMR National Institute for Research in Reproductive and Child Health, Mumbai, and Kasturba Health Society Medical Research Center, Mumbai, to Professor Dr. Gagandeep Kang. It reads as follows. Dr. Gagandeep Kang, a renowned medical scientist and a distinguished professor, is currently the Director Enterics, Diagnostics, Genomics, and Epidemiology, Global Health at Bill and Melinda Gates Foundation, USA, and Adjunct Professor at Christian Medical College, Vellore. She has served as the Executive Director of Translational Health Science and Technology Institute, THSTI. Dr. Kang's interdisciplinary research demonstrated the high burden of rotavirus disease in India, the genetic diversity of viruses, and the factors that affect transmission of disease in hospital and community settings. She has set up national surveillance network, established laboratories to support vaccine trials, and conducted phase 1 to 3 studies of rotavirus vaccines in partnership with non-government and government organizations. She has developed a strong training program for medical students and young researchers in clinical translational medicine with an aim to build next generation of clinical researchers to address India-specific health issues. She has published more than 400 research articles in national and international journals. Dr. Kang has received several accolades, including the Infosys Prize in Life Sciences in 2016 and the distinction of being the first Indian woman elected as a Fellow of the Royal Society in 2019. She is Fellow of all the 3 science academies in India and also a member of the US National Academy of Medicine. This citation is being presented to Professor Dr. Gagandeep Kang in recognition of her outstanding contributions in the area of public health, infectious diseases, and vaccine development. Thank you.
Thank you, ma'am. It is a privilege to have Dr. Gagandeep Kang with us. She has joined us online. So we request her to take the stage and share her expertise on the topic building capital, human capital in India, cohort studies, nutrition, and need for collaborative science. Welcome, ma'am.
Thank you very much for the invitation to join you here today. Are you able to see me?
Yes, ma'am. Very clearly, we're able to see you.
Okay. I'm going to be sharing my screen. Please let me know when you can see that.
Yeah, ma'am.
Okay. Thank you very much. I'd like to thank Dr. Gitanjali Sachdev for inviting me to deliver this oration. Professor Vandana Patrawale for the outstanding address in her role as chief guest, but also all our other colleagues from the ICMR National Institute for Research in Reproductive and Child Health and the Kasturba Health Society Medical Research Center. Particularly, I'd like to thank the family of Professor P.K. Devi for joining us. I was very pleased to learn that one of my colleagues, Nitya Nair, is related to Professor Devi. So it just goes to show what a strong woman scientist can do, as was shared by Dr. Jayanti, around the country and the institution, but also within her family all as well. When I was looking at the people who had given this lecture before and thinking about what to talk about, usually I speak on vaccines, but then given Professor Devi's expertise and contributions to reproductive and therefore child health, I thought it should be structured somewhat differently. So what I'd like to talk to you about today is our learnings from the studies that we have done over the last 25 years, and then think about what might be a path for us to go forward. Now, as everyone is probably aware, I'm a virologist, and I started out with laboratory studies of enteric infections. So how do you make a diagnosis? Can you build animal models of disease? But I very rapidly transitioned to thinking about the role of these viruses in public health and to thinking about what kinds of interventions we could have for our children. Group A rotaviruses are the most common cause of dehydrating gastroenteritis in children, and it is an unfortunate fact that the children that die of dehydration are the children that cannot access health care. So therefore, it was very important for us to think about prevention of disease rather than thinking about treatment alone. And there are many ways, obviously, to prevent enteric infections. However, it is an unfortunate fact that diarrheal deaths continue to be a problem, particularly in South Asia and in sub-Saharan Africa. If we look at the figures that come from the Global Burden of Disease studies, as late as 2019, 6.6% of children under the age of 5 who die in India, their deaths can be attributed to diarrhea. The percentages, of course, are much worse in sub-Saharan Africa. So while we have made a lot of progress, we are not really where we should be in making sure that our children do not die of a preventable disease. So working towards this, I was in both in Vellore and during a postdoctoral period in the US, looking at rotavirus vaccines as a road to prevention. Now, while I was in the US in the 1990s, a new vaccine was licensed. It was called RotaShield. It was the first licensed rotavirus vaccine. It came about 25 years after rotavirus had been identified as a cause of diarrhea in children. It was first licensed in the US, which is kind of unusual because you wouldn't expect a vaccine that is for a diarrheal disease to be licensed in a high-income country. But we forget that while there might not be deaths due to diarrhea in the US, there are certainly a lot of hospitalizations. So the US, when this vaccine was licensed, introduced it very, very quickly with 3 doses of this oral vaccine to be given at 2, 4, and 6 months. This was data that was based on a trial in over 10,000 children. Unfortunately, this wonderful vaccine, which had been developed by the National Institutes of Health, had a side effect. It had very good protection against severe gastroenteritis, but after 1.5 million doses of this vaccine had been given, it was reported that there was a possible association with intussusception, where one part of the intestine telescopes into another, and the vaccine was withdrawn. It was noted that these kinds of adverse events happened in the first week after the first dose of vaccine, an increase in cases of intussusception just after the first dose. After the second dose, there was a small signal and nothing seen after the third dose. Now, in the US, they withdrew this vaccine. And in India at the time, we were just preparing for a trial of this RotaShield vaccine. So since that vaccine was withdrawn, we then followed another path, which was the development of an indigenous vaccine candidate. But while those studies were being done of the indigenous vaccine candidate, we decided to work with collaborators in the US and in the UK to try and identify what would be a correlate of protection for the vaccine. At that time, it was unknown, and sadly, it is still unknown how a rotavirus vaccine protects against diarrhea. So in order to do this study, we decided that we would establish a birth cohort and that we would work in an urban slum area in Vellore. And this map shows you the houses of the various participants. You can see that they're quite closely clustered together. Now, this was an area of approximately 2.5 square kilometers, and our cohort study design was to have very intensive follow-up. So we recruited mothers when they were pregnant and waited until their children were born. We collected cord blood, we collected a first stool sample, and then we did surveillance visits twice a week, collecting stool once in 2 weeks, investigated all diarrheas in children and their families, collected sera for every rotaviral episode, characterized the strains, characterized the immune responses, and then looked to see how this correlated with clinical information. This was the first spatially enabled database that we had built for a cohort study. This is what our area looked like. As you can see, there was really no shortage of children in the community. So we were able to recruit almost 400 children from a very small area and then follow them up. So when we completed follow-up for the cohort 3 years later, we had followed 373 children for about 13,000 child months, and we had collected 27,000 stool samples for surveillance, almost 5,000 for diarrheal episodes, about 4,000 serum samples, so averaging about 11 samples, 11 to 12 samples per child. And we had evaluated all of these samples by ELISA, by PCR, and by immunoassays to detect antibodies to rotavirus. Now, we did learn a lot from this study. We learned that in India, children are infected very early. About 50% of children were infected in the first 6 months, and by the time they were 36 months old, almost all children were infected. However, only about half of the children had a detectable rotavirus gastroenteritis in the study design that we had set up. Now, these data were very useful because nobody does this kind of detailed community study, and we were able to use this and data that we were collecting in the hospital to estimate a disease burden of rotavirus in India. And what it told us was that about 1 in every 650 children born in India would die of a rotavirus gastroenteritis. 1 in 30 would require hospitalization. 1 in 8 would need an outpatient visit. And 1 in 2 children would have a diarrheal episode. So while we were doing all of these studies, we were also measuring growth in these children. I'd like you to look at the black on the top of the figure. That is a normal growth, and it was seen in 28% of the boys and 37% of the girls that we studied. If we looked at the remainder of the children, so 70% of boys and a little over 60% of the girls, they had some form of growth faltering. Not a lot of wasting in our community, but a lot of stunting and underweight children. Now, this was interesting to us because the data that we had actually replicated studies that had been done in South America in a cohort by Leonardo Mata. The monograph has been published as The Children of Santa Maria Cake, and it informs nutrition studies around the world. What they did there was to plot what a normal growth curve would look like, and then document what taking various hits because of intestinal infection or other infections, what that did to the growth of children. So if you look at it, they were studying episodes of bronchitis, bronchopneumonia, cellulitis, conjunctivitis, diarrhea, fever, impetigo, measles, etc. And what you can see here is that for a child, having these repeated episodes of illness essentially means that once you fall off the growth curve, you stay off the growth curve. And this can be lead to a lot of damage. So I'll give you an example of a child with what is called a matagram after Leonardo Mata. What does it look like for a child with enteric infections? And here I am only showing one particular parasite that we studied, Cryptosporidium. What you can see here is a child that was on the growth curve initially and then fell off and stayed below the normal growth trajectory. Now, she's not very badly off. What we do in our clinic is to use red and green tapes to show where a child should be, and then red constitutes an HAZ, which is a median of less than 2 standard deviations. So this is actually Parvati. And Parvati had a lot of risk factors going for her. She was born low birth weight. Her mother had no formal education. She lives with an extended family and siblings. They have no toilets and they keep poultry. Now, the one good thing that you could say is Parvati was exclusively breastfed for at least 6 months. Now, the problem with that kind of approach to looking at growth is you can see Parvati's mother. And one of the reasons that a child is exclusively breastfed for a longer period of time is because the mother has very little that she can give to the child other than breast milk. So, children in this environment don't always grow very well. This is an environment that we are all familiar with. It is a setup for children to have repeated enteric infections. So we became interested from studying the correlates of protection. We became interested in what living in this environment does to children. And we started to look at sampling their environment. What we found, obviously, was that there were very, very high levels of contamination in everything that we tested, in the food, in the water, in the surfaces on their household. This was not really different from what had been seen in multiple other studies around the world that had been looking at diarrhea and what the causes of diarrhea were. In our study, as well as in the GEMS study, which included countries in Asia and Africa, we showed that rotavirus was the #1 cause of diarrhea in children during infancy. There were other causes that came afterwards. Cryptosporidium, which we followed for Parvati, was #3 if we looked at which pathogens were causing enteric infections in children. Now, in these studies, we looked at both children who had diarrhea as well as asymptomatic infections in these children. And we found that children were carrying pathogens pretty much all the time. Now, what does it mean to have an infection? We are familiar from histopathology with the finger-like villi in the gut that contribute to a very large absorptive surface. However, when you get an infection, what happens is that the rotavirus infects the mature enterocytes at the tops of the villi, and that results within 24 to 72 hours after infection in essentially a complete blunting of the villi. This means that you're actually losing absorptive capacity in the gut during the period of infection. There is inflammation, there is infiltration of white blood cells into the lamina propria, as well as vacuolation of epithelial cells. Now, rotavirus is a relatively self-limited infection, and usually recovery happens within 1 or 2 weeks. However, if we look at other enteric pathogens, they have different mechanisms of causing damage to the gut, but all of them result in a gut that is damaged, that is unable to absorb nutrients and unable to maintain its barrier function. So these kinds of effects can have profound consequences for children. Repeated hits because of infection result in a condition called environmental enteropathy, What you see here in the first column is healthy controls, and next to that is children with environmental enteropathy, where you can see that the damage to the gut is as much as the damage that you see because of other more familiar conditions that include things like celiac disease. So this environmental enteropathy can influence how the gut functions. And we decided in our community to look at what the environment really means. One of the things that is very rarely studied, we talk about environment, we talk about developing countries, we talk about, the fact that people have lots of infections. But I think it is really the built environment that makes a difference. And we were able to show this by looking at children that lived in the urban slums that I've shown you and comparing them to other children in Vellore, which were essentially the children of doctors who worked in CMC. What we were able to show there is that it wasn't like the children of doctors in CMC had absolutely no pathogens at all. If you sampled a perfectly normal asymptomatic child from the CMC campus, you would find one enteric pathogen in their gut on average. But if you went to the urban slum and you sampled children there, then you found that they had 4 times the number of pathogens that children from a higher socioeconomic status had. Now, having a pathogen in the gut can actually lead to a lot of effects on permeability, damage, translocation, inflammation, etc. And there are many ways to measure this. In many of the studies that we've done, we've used practically at least one of the markers from each of these categories. I'll just show you one example, which is calprotectin. You can see that CMC doctors' children have relatively low levels of inflammation, whereas children from the EVI study were children who had very, very high levels of inflammation. In fact, this level of inflammation in children who are apparently normal is higher than the level of calprotectin that you would see in inflammatory bowel disease. So imagine what living with this level of inflammation in your gut over years can do to you. So we've been studying these complex interactions between entropathogens and diarrhea, entropathy, nutrition, growth, cognition, and wash behaviors. So in order to do that, we have followed many cohorts over long periods of time. We are now at over 20 years of follow-up in one of our first birth cohorts. And we have then had another 4 additional birth cohorts that we have recruited since then, where now the youngest children are over 10 years of age. Now, in the first cohort, we started to examine not only growth, but also to look at the cognitive performance of these children. And what we found was that when you expect the average IQ of children to be somewhere between 90 to 109, only 1/3 of our children were average. Only one child was high average. The bulk of our children were in the low average of 80 to 89 IQ, and the rest were lower. We tried to figure out what were the factors that contributed to this, as has been shown in many studies before. IQ and the home environment are associated with what happens to children. So maternal education and socioeconomic class played a role. But if you looked at stunting, you found that if you had children who were persistently stunted, they had lower IQs than children who were not persistently stunted or never stunted. Now, when we moved from a simple regression to a multiple linear regression, it still held up that prevalence of stunting and maternal education and helminthic helminthic infections were contributing to lower cognitive performance in later childhood. So remember, these measurements are taken when the child is 1 to 3 years of age. These children are followed up at 7 years, 12 years, 18 years, etc. And then we characterize children as being persistently stunted or catching up. So while catching up, there does appear to be a dose response effect, where if you catch up, the damage is less, but if you're persistently stunted, the damage is greater. And in measurements that are done at different time points, you can see that the correlation continues to exist. So overall, where we are today, we know that children in our urban slums in southern India now have 30% stunting as opposed to the 60% when we first started out. The median IQ in our slums is about 89, which is low normal. And persistently stunted children have lower IQs than children who have never been stunted. So this was followed up by a developmental pediatrician at CMC, Beena Koshi, and she has been looking at both the original cohort as well as an additional cohort to look at what happens to these children using now new tools. So she's been using MRI brain volumetry to analyze what persistent stunting does to children. I won't go into details of this because this is not my area of expertise, but essentially her analysis found that if you look at gray matter, there is no major difference between stunted and non-stunted children. But if you look at the cerebellum and the corpus callosum, what you find is that the volumes are smaller in stunted children. And this has effects for behavioral and intellectual function, including for language integration. So when you look at this, it's very important for information integration as well as for motor coordination. Beena is continuing this work, and she's also beginning to work with other colleagues on more detailed investigations of these children. Now, these data are from Vellore. What about the rest of the world? If we look at acute and chronic malnutrition, the most recent figures indicate that about 150 million, less than 5%, less than 5 years are stunted. And this is 32% currently of Indian children. And if we look at wasting, 45 million children are wasted and 7% of children in India are wasted. Now, this global disease burden, if we look at it, you can see that the AIDS standardized prevalence rate of protein energy malnutrition is shown in the top graph. It's not a good thing to be in this category. If we look at changes that have been happening, there is obviously some change in India shown in figure B, but it's not nearly as fast as we would like it to be. Now, we have had Poshan Abhiyan, which is a package of interventions that is delivered from pre-pregnancy all the way to 2 years of age. There are many things that are being done by different stakeholders within the system. These range from wash to feeding interventions, both in outpatient facilities and, if required, in inpatient facilities. But the needle hasn't really moved as much as we would like. what we saw was that in the most recent World Bank review, the messages were getting out, but the outcomes were less than had been expected. So in terms of looking at a minimum acceptable diet by children between 6 and 23 months, only less than 20% of children actually had access to that. So if we look at NFHS and our data, there has been little change in the past decade. There are a few things that we know, that stunting is affected by low birth weight, it is intergenerational, and low SES can make a difference. If we look at wasting and thinness, that can be a precursor to later stunting. What does this mean for interventions? Some of the things have been incorporated into what has been laid down as policy now, particularly with Poshan Abhiyan, but implementation continues to be somewhat of an issue. We know that we need to support breastfeeding, but we need to start actions early, including in adolescent women and in newlyweds. We need to make sure that there is family support, and we need to look at the quality of complementary foods and the challenges that families are facing in ensuring care for mothers and babies, particularly in the first 1,000 days. So where should all of this work be done? If you take anemia as a measure of maternal nutrition, you can look at this data from NFHS 5 plotted by S.V. Subramaniam at the Harvard University. And what you see here in the first graph is the levels of anemia, and you see it in the north and the east, it is the greatest. But if you look at where change has happened, you can see that the least change in the green and red map is in central and western India. So these are areas where looking at maternal nutrition is important. And if we come to children, we see that stunting in children in NFHS 5 is again in central and parts of northern India. Where change has happened between NFHS 4 and 5, 2016 to 2021, is fortunately in at least some of those areas. And we need to think about how else we implement better in those regions. So essentially, while we've made some progress, business as usual hasn't solved our problems. So we have to think about what are the scientific and technological interventions that we need to have in order for us to achieve our targets and global targets. Our current trajectory is shown in blue, and that's why we need new science to allow us to do better. And we need to think about how we measure the impact of our interventions, not just reduction in severe acute malnutrition, not just reduction in stunting or in anemia or micronutrient deficiencies. We need to be looking at indices of development, the Human Capital Index and the Human Development Index. We now have many tools available to us to study gut function. There are many kinds of tests that we have begun to apply. And it is important that we do this because if we are to bring new science to solving an old problem, then it does require new tools that we should be using for this. One of the areas, as Dr. Vandana had pointed out, is microbiome. And I was pleased to be part of a study across India, Bangladesh, and Africa, where we were able to identify a limited number of microorganisms where there was an association of severe acute malnutrition, and we could think about how to modify the microbiome with therapeutic foods that are designed to target the microbiota so that children, by just eating food, can repair their guts and have better nutrition. So there are, when we think about a nutrition science research study, we are going to need collaboration, but within India, we have a lot of strengths. We have people who are skilled in basic translational, clinical, and public health research. We have unique animal models, patient cohorts, and biobanks that we can use for metabolic phenotyping. We have links to the program. We have multidisciplinary scientists. So we can look not just at stunting, wasting, and anemia, which affect how you develop, but we can also think about going beyond them to look at the double burden or metabolic syndrome, look at aging, inflammatory disorders, and even cancer to look at how better to feed patients. So there is a new group at TIFR Hyderabad that is led by Ulas Koltur and Mahendra Sonavane. They are building a model for collaboration in India, looking at alleviating malnutrition and countering the double burden of malnutrition. These are basic scientists that have now linked up with multiple institutions across the country to be able to use clinical information to inform their development of model systems. The idea is to understand the biology of malnutrition so that we can think about mitigating resistance to the feeding programs, where feeding programs don't work. How do you solve that? How do you maintain physiological fitness and move beyond just anthropometry to measure what is happening in both children, adolescents, and adults? So the idea here is to use the tools we have, which are human and animal biopsies and organoids, to develop cellular models, to think about how you develop animal models and use human cohorts to look at the gut microbiome, potential dietary interventions, and study genetic variation, and also look at molecular and cellular mechanisms that inform energy expenditure by looking at mitochondria, the metabolome, as well as using phenomics and genomics. The idea is to have truly informed human interventions as well as biomarkers that can predict outcomes and give us an understanding of long-term implications of inappropriate interventions. We know very well that just supplementing calories is no longer enough. You need a balanced energy product to be able to deliver on malnutrition. So they've established a number of platforms, stable isotope methods, imaging analytics, metabolomic platforms as well. They have a national analytic facility that is across TIFR and BARC. And they are looking also for bringing to India for the very first time at across TIFR and CMC Vellore, human metabolic chambers where you can actually conduct experimental medicine on humans, where you're fed a labeled diet and you study energy expenditure and can measure input and output. So just to end, building our human capital really requires changing the nutritional status of our children. If our position on the human capital index is to become a much darker blue, there is a lot that we need to be thinking about, and that requires for us to think about children, their mothers, the gut environment, the external environment, and what we can do to influence that through water and sanitation, antimicrobials and vaccines, nutrients, and the microflora that we all live with. If we are to change the human capital index and to bring everyone in India out of poverty through improving their cognition and physical well being, then we do all need to work together. Thank you so much for your attention.
Thank you, ma'am. We are indeed honored to get an opportunity to listen to the extensive work you've done in public health and through this oration. Now is the time for the Oration Award presentation. What is an e-oration, and ma'am will be presenting it to you virtually. And for your, I invite our director, ma'am, Dr. Xxxii Prof, and Dr. Devi, ma'am, for the handing over the Oration Award to Dr. Gagandeep Kang for her outstanding work in public health, infectious diseases, and vaccine development. We can just, ma'am, yeah, yes. Here we can see that camera we can use the screen there.
Thank you.
We are thankful to the NIRH team for hall arrangements, logistics, catering, and administrative support. And finally, we thank the delightful audience present physically and virtually for making this program a great success. Before I end, I request everyone to rise for national anthem. Thank you, everyone.
तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
When you write, your tone influences.
I would request for a group photograph for all the guests, the program organizers, team from KHS MRC, and all the faculty members from NIRC. Please join us for the group photo. We have also the refreshments served outside. So guests who are attending online, please stay online for the group photograph and please switch on your video. Yes, ma'am. Please switch on your video, whoever. Yes, ma'am. Thank you so much. Okay.
Are we done now?
Okay. So thank you all who have joined online for the e-oration. Thank you, Gagandeep ma'am. Thank you, sir. Thank you.
Thank you.
Bye. Bye.